Okay, everybody. Well, welcome, everybody, and thank you for taking time on your Saturday, or in some places it might be your Sunday. I am John Hopper, the president of the Fibrolamellar Cancer Foundation, and this is our virtual research series that we call the Evolution of Research and Clinical Care in Fibrolamellar. Today is an open house with the University of Washington, which I think you will find extremely informative. Hey, importantly, in the world of virtual, we're now able to connect each other across the globe. Today, we have people from 4 continents. And again, thank you for all attending. This is an extension of something we started in 2016 about getting patients, caregivers, and researchers together so that we can bond and learn from each other. And we've been doing this since 2016. We've actually done this at the Mayo Clinic, at Mass General, at Duke, at Cornell, University of Washington. I think some of our friends today who are on here, like Tom Stockwell and Tracy O, the Browns, the McDonoughs, the Greggs, the Berries, the Greens have all participated in this. So I'm glad everybody can take part today. We have a couple of other upcoming events, so please go to our website to sign in. In February, we have on the 25th, a panel discussion on clinical treatments with a who's who of clinicians on fibrolamellar. In April, we will have one on clinical trials. The date's still TBD, but again, check the website. And then in November, for the first time ever, we are able to have an international research conference, November 1st through 3rd, where patients, caregivers, and researchers will gather together. Again, check the website for more to come. I do have to acknowledge a few things. One is the Chan Zuckerberg Initiative was able to give us funding to do this series of programs, as well as Bristol Myers Squibb. And I would be remiss not to thank my team at the FCF, Lynn O'Malley, Megan McKinney, Sandy Vosses, Kurt Losart, Mark Firth, and Jenny Carroll. So thank you all for putting this together. For those of you who are new to the Fibrolamellar Cancer Foundation, just a quick 2 slides here. One is we were started in 2009 by fibrofighter Tucker Davis and his 3 friends. Tucker's parents, Martin and Chuck, have taken the torch and continued the foundation with a 3-fold mission. The first is to find a cure and treatment options for fibrolamellar. It's to raise awareness of this disease, and also to connect and support the fibrolamellar community of patients and families. And since Tucker started this, you can see on the bottom the logos of very prestigious institutions that have basically come around to support the fibrolamellar community. So thank you, Tucker. Since our inception, we've also invested over $8 million across 24 institutions. I won't read them all for you right now, but the key thing here is no one gets $1 from us unless they commit to collaborating with each other and sharing data. And importantly, every dollar raised goes to research because Chuck and Marna help to cover our operating costs. So again, thank you everybody who's participating in this. And again, collaboration and shared data is essential to find a cure. So today, it's our virtual research open house. We call it Bench to Bedside, with a very prestigious University of Washington. The University of Washington to us was very unique because it has a multidisciplinary approach to fibrolamellar research. As you can see here, it's the University of Washington Medical School, it's the Seattle Children's Group, Seattle Cancer Care Alliance, and Fred Hutch. And again, we've been so impressed with these folks that we have funded over $700,000 in University of Washington research. And this team here actually has won over $1.5 million from the Department of Defense, which is an extremely competitive field out there. It just shows their commitment to fibrolamellar. Today's hosts, I'm very proud to say, are the distinguished Dr. John Scott. John is the Edwin G. Krebs-Speights Professor of Cell Signaling and Cancer Biology, and he's the chair of the Department of Pharmacology. We're also pleased to have a co-host, which is a fibrofighter since 2004, Mr. David Burch. David, many of you already know, very active in the community. and we consider him one of our Fibrolamellar community ambassadors. So our agenda today, so I can hand it on to the people you really want to hear from, is an overview of the University of Washington's Fibrolamellar focus. We're going to hear the patient journey from David. We have two panel discussions. One's on the clinical pathways and the other one's on the research universe. And if you want to participate, at the bottom of your screen, you should see a Q&A. Just click that on and then pose a question there. And then our own Dr. Mark Firth will actually help to moderate those questions. And at the end, all I can say is if you have any inquiries after today, if any of your questions weren't answered, just email us at info@fibrofoundation.org. And with that, I am going to turn it over to John as soon as I can stop sharing my screen. Here we go, gang.
Okay, thank you, John. And as we've become used to with the recent years for presentation, good morning, good afternoon, and good evening, wherever you are. It's really my pleasure to sort of give an introduction to our team here in Seattle. And I'm going to do that by beginning with a presentation that I've got here that I'm just about to pull up. And just really start with some of the questions that we think are important to consider as we begin to talk about this today. So clearly, you know, once somebody is diagnosed with a disease such as fibrolamellar, there are so many questions that they face. And these questions include things like the clinical, how they're going to be treated, the trust of their physician, and possibly being involved in trials, clinical trials and research. And it's really as we transition from the care to the research that we're going to focus on today. And also on the other side of that balance is what happens in a clinical medical center such as the University of Washington and Children's Hospitals, where we are all laser focused on cancer, but there are different elements and aspects of this that we also need to consider, which are how are we going to get the best care, what are the right treatments to be used, and what are the research options. And it's really within that context that I'm just going to do something here for a second. Sorry. It's really within that context that we are able to work towards what we're talking about today. So this really is a way to sort of introduce our group. So we are really incredibly happy to be supported by the Fibrolamellar Foundation. These are people that support our science, but they are also people we respect immeasurably, and they've become our friends. And this is a unique collaboration between UW Medicine, University of Washington Medicine, Seattle Children's Hospital. And we're all physically located in the Pacific Northwest. This is a picture here of the University of Washington campus. And you can see the hospitals, the University of Washington Medical Center and Children's Hospital. And our focus really is on this idea that it begins with the patient. It really begins with the care on the clinical side, and then it moves towards research. And so we've come up with this term, which is really from the bedside to the bench and back. And that's the kind of focus that we're going to have today. We realize that it begins with clinical care, and many patients are then asked if they will participate in research projects. And they're usually very enthusiastic and excited to do that. But it's when we do that research that we can then give back to the patients by coming up with innovative and better forms of care and treatments. And that's what we're going to talk about today. And so that begins in a hospital and it ends up in a laboratory, and then it goes back. So there's a short video here that shows you how this happens at the University of Washington. But this is the doors to the hospital. And we have a lot of very long corridors where we take our samples, sometimes straight from the OR to a research lab. And it's a very different environment, but it's an important part of this whole process where we can begin to analyze the samples we can begin to investigate new avenues towards being able to treat this disease and come up with better therapies and maybe ultimately a cure. So you'll be hearing from Sophia later on in this presentation, but this is just a picture here of her getting cells ready to begin to look at what is going on in fibrolamellar. Now, this machine that looks like an ice maker is actually a $1.5 million super microscope that allows you to look at these cancer cells in real time while they're still alive, as you can see here. You can actually watch what's going on in these cells, and by observing, you can learn a lot. So that gives you a sense of how the research goes forward. And I'd also like to just finally end this part by introducing our team. As John said, it's a very multidisciplinary group. We have surgeons in Kimberly, Riehle and Raymond Yeung, you'll hear from them, and Venu Pillarisetty, who are surgeons. We have primary care clinicians such as Bill Harris and research fellows, Alan. And then there's the more straight researchers such as Turan Gurjal, Sophia Lauer, and myself. We're also spread over really 3 world-class institutions, the University of Washington, the Fred Hutch Cancer Center, and Children's Hospital. So I hope you enjoy our time together. I am now going to pass the baton on to David Burch, who is one of our patient care ambassadors, and he is going to guide you through the process. So I will talk to you again a little bit later. So David.
Thank you, John, and hello to all of you. Like both of the Johns had mentioned, my name is David Burch. I've been a fibro fighter now for 16 years, and I really have no plans to stop. I'm here to talk a little bit about my experiences and what it's like at a medical facility like the University of Washington, but also to share a bit of my story. I wanted to start with my background. When I was 22, I was taken to an ER close to my home for some excruciating pain in my side. This turned out to be a softball-sized lump in my liver that would be removed 2 days later. This would eventually be diagnosed as fibrolamellar hepatocellular carcinoma, but it was originally diagnosed a different type of liver cancer. After it was removed and things started to settle down, I did find that dealing with the diagnosis at times was way worse than the actual cancer itself. When I was diagnosed, I felt shamed in the questionings and accusations that were asked of me. I was 22 years old and was being asked repeatedly how much alcohol I drank, what kind of sexual experiences I had. At the time, it was thought that the only way to get liver cancer was by having cirrhosis of the liver or having hepatitis A or B, or at least that's how it felt. And it was really nonstop for a while. I still remember a time when a doctor flat out told me that I was lying to him about some of the things I was saying. It wasn't until I needed my first lung surgery, which was a year later at the University of Washington, that I was correctly diagnosed with FHC and was able to start getting the treatment that I needed. This, however, was all before I ever came to Seattle Cancer Care Alliance, or the SCCA for short. It wasn't until 2012 that I met with now my primary oncologist, Dr. Bill Harris, who is also actually on the panel today. I didn't realize at the time that UW and SCCA worked together. And after my first few visits with Dr. Harris and with some unfortunate dealings with my local clinic, I decided that I would transfer my care from the local clinic to Dr. Harris, along with SCCA and the UW. Immediately after I started going to SCCA, I knew that it was the right place for me. Within the first few visits, I knew more about my cancer than I ever knew while I was going to the local clinics. And even though the drive was painful at times, about an hour each way, not including any horrible Seattle traffic, it was well worth it. It felt refreshing to go to a place where I did not feel like just another statistic. I started finding out about clinical studies and different ways to treat the disease. Bill Harris was also the one that told me about a little organization that was starting to fund and help with my kind of cancer, the FCF. One of the most fascinating things I have been a part of since moving my treatment from UW to SCCA was that in 2018, my wife and I were invited to a seminar and Q&A session, a lot like this one. The main differences were that I actually got to see everybody and meet everybody in person, but also we didn't have to wear masks. It was there that I actually saw that I learned a lot of things like where tissue samples went, what new treatments were being looked at, and even that they had found similar things in all the different samples. One of the things I remember is feeling very encouraged with this. I wanted to do more. I wanted to see how I could help. This is a big turnaround from where I was when I was first diagnosed. I remember being told at one point that I probably only had 2-3 years maybe to live. I was told that this cancer was a death sentence and they would never find a cure. I was told that due to certain chemos that I was on, that I'd probably not have kids. I do have a side note. I do have now a 4-year-old girl and a 7-year-old boy and an amazing wife. But hearing from the doctors and scientists at the seminar in 2018, some who are actually here today, gave me what I didn't think I ever had, which is hope. I know that there is still a lot that needs to be done, but I felt the drive to want to do more. I wanted to do as much as I could to help, and it even pushed me to make myself healthy again, to where I was able to complete one of my goals since getting cancer, which was to do a 7.5-mile road race that I had not done since high school. Even though it is now about 3 years later and I've had more surgeries, radiation treatments, and some side effects that I wish I didn't have, I am still just as driven now to help. But if there's one thing I really want anyone here at home to get out of this research open house, the same thing that I got out of it, which was hope. It may feel hopeless at times, but know there are people out there who are looking for a cure. With all that being said, I want to introduce the next couple people on the panel, Dr. Bill Harris and Kimberly Riehle.
Thanks very much, David. It's great to hear your story. So can you guys hear me okay? Yeah, okay. I'm Kim Riehle. I'm a surgeon at Seattle Children's Hospital, and I'm very pleased to be here today to talk about the collaboration throughout the institutions in Seattle, and to again thank the Fibrolamellar Cancer Foundation for their support and really how they have elevated the level of interest in studying this cancer across the country by their advocacy. So today, Bill and I are just going to talk through some basics about how patients are introduced to us and get their diagnosis and how we institute treatment. So as many of you know, and as David alluded to, patients who get fibrolamellar carcinoma are otherwise completely healthy. They're not like other liver cancer patients who have chronic liver disease. And so they usually present with very subtle symptoms. And in kids, I feel like they mostly come through our emergency department because suddenly someone has developed pain or maybe feels a mass. And as a parent, that's obviously very anxiety provoking. And so most people don't want to wait for a clinic visit. They just bring their kids to the ER. And so in children, we will usually start with an ultrasound to try to get a sense of where the mass is coming from. And then we'll send off some basic blood work to see whether the liver is functioning okay, which in a fibrolamellar patient, we would expect that it would be, and then set up some more advanced imaging. In fibrolamellar patients, as most of you probably know, the labs would be completely normal. There's a tumor marker called alpha fetoprotein, or AFP, that can be elevated in other types of liver cancers, especially in children, but this is normal in patients with fibrolamellar. And you can see here in this picture that ultrasounds are a little bit hard to interpret in terms of what exactly is going on and where the tumor exactly is, which is why we then get more advanced imaging tests. And usually in the emergency department, the faster thing to do is a CAT scan. And so down in the right lower corner here, you can see a patient who has a fibrolamellar carcinoma. And it's this mass here. And in the center area, there's an area that's not quite as bright, and that's what's called the central scar. And if you look at that area under the microscope, that's where you'll see those fibrolamellar bands, which is how this tumor got its name. But there is one other tumor that can look just like this on a CAT scan, and that's called focal nodular hyperplasia. The name isn't too important other than that is a benign tumor and can sometimes be observed. And so it's really important to try to tell the difference between a fibrolamellar carcinoma and a focal nodular hyperplasia. And that's why most patients will then get an MRI to try to really distinguish between the two. And the big difference on the MRI is in this third panel here, where a special type of contrast is given called EAVIST or some other type of what we call biliary contrast. And a cancerous tumor will not take up that contrast, whereas the benign tumors will. And so, that can often really help us counsel the patient as to what we think is going on. And then if there's any question after the MRI, a patient might get a needle biopsy, but I've found that most of the time our radiologists are so good that they can make the call based on the MRI, and then we can talk about what to do. And as many of you know, really the best treatment for fibrolamellar continues to be surgery. And so then we really try to think about, can we do the surgery safely? And so the way we do that, and this is kind of a complex topic that I'll just try to summarize quickly, is we use the imaging to try to figure out how much of the liver is involved, mostly because we want to make sure that when we do the surgery, we're leaving behind a healthy remnant liver. And so we really think a lot about the liver we're leaving behind. The liver is really an amazing organ in that it can grow back after you remove a big section of it, but it really has to have good blood flow going in and good blood flow coming out. And so we spend a lot of time really assessing those blood vessels and making sure that we can safely get the tumor out without damaging the liver we're leaving behind. The other thing that's really important about fibrolamellar carcinomas in particular compared to other liver tumors is that we really want to assess whether the lymph nodes in the area have any tumor in them. And so that's a really important part of the surgery. Now, unfortunately, in some patients, again, because they're otherwise so healthy, some patients present with disease that's not amenable to a traditional surgical approach. In this upper picture, here's the main tumor, but then the blood vessel that's right below it has some tumor within it. And so that's a patient where you'd either have to think about getting that tumor out of the blood vessel, maybe reconstructing the blood vessels, or in this lower patient here where you have multiple nodules of tumor, if the part you're leaving behind is not going to be adequate, that might be a patient you would think about referring for a transplant. There are also some techniques that are done by our interventional radiology friends where you can try to deliver radiation to a specific part of the tumor, or other methods to try to locally shrink it and allow the other part of the liver to grow a little bit in order to try to do the surgery safely.
Great. Thank you, Kim. And so my name is Bill Harris, and I'm a medical oncologist, and I am on faculty at University of Washington, but my primary clinic is at Seattle Cancer Care Alliance. And I focus on treating patients with liver or bile duct cancers, and tend to see patients in the Northwest who are diagnosed with fibrolamellar. So I tend to get involved from the medical oncology side for essentially all of our patients. I'm at least approached and aware of them. And I'll just say that this is a very multidisciplinary approach to treating patients. And it takes a big team, as I think you'll get the gist of from this presentation. When we talk about patients, what happens after surgery, a couple things I will highlight are that, there have been advances in understanding fibrolamellar, and this gets to the fact that we really need to integrate our current knowledge from research with our clinical care. And one example of this is that it's become recognized that the vast majority of patients with fibrolamellar have a specific mutation within their tumor. And so we do recommend getting genetic testing on the tumor itself for patients with fibrolamellar. And one example is that this mutation highlighted here, which puts two different genes, normally which are separated in juxtaposition has been recognized as extremely common in fibrolamellar. And we have now integrated that testing for that mutation into our normal genetic testing panels through our University of Washington lab medicine tests. So we can now identify this mutation along with looking for other potentially relevant mutations when we extract DNA from a tumor and analyze it. In terms of what happens after surgery, hopefully the surgery was successful in clearing all visible sites of disease on scans. But it's important to continue with careful imaging and surveillance. And often I work with our surgeons in multidisciplinary group, and we assign a point person to take over surveillance. And usually we do surveillance more frequently early on. As Kim mentioned, that could include MRI, but it can also include other scans depending on the exact situation. And when we see findings where we're not quite sure what to make of it, we often will take that back to a multidisciplinary group, including surgeons and liver radiologists and myself and others, just to figure out what the best next steps are through a consensus of many specialists who have experience in fibrolamellar. Eventually, if things are going well, we will transition out to less frequent surveillance, but we continue to follow people quite long-term for this. Next slide, please. So if fibrolamellar does come back, it's important to understand that repeat surgical resection is certainly appealing if it's possible. And so again, that gets to an extensive discussion with our multidisciplinary groups. If we can't do repeat surgery, then there are a lot of other options, and we call them local therapies to treat specific tumors. And so this gets to the fact that you need a broad expertise at an institution. And that can include things like our interventional radiologists who can do procedures to treat specific tumors, or our radiation oncologists who can use advanced radiation techniques to treat metastases if it is spread away from the liver itself or recurred within the liver. And then chemotherapy and other drugs, and I'll sort of lump that into the term systemic therapy. And that can include chemotherapy, immunotherapy, or clinical trials, which I'll touch on in a bit. Next slide. So I do want to highlight some of our experience, just noting that if the cancer does recur within the liver or away from the liver, we do circle back to see, can we treat this, with local techniques? And we have patients who have been treated for many, many years and even decades with primarily going back and treating those specific tumors. Of course, we consider surgery, but there are things like ablation of the liver or lung lesions, where we can stick a needle in and essentially burn those sites of disease with good result. We have very good radiation oncologists and have had good experience controlling tumors with radiation for many, many years without evidence of recurrence at the site that we treat. And then our interventional radiologists can do things like arterially directed therapies, where they can put a catheter into the blood vessel that feeds these tumors and release things like radioactive beads to treat, often in this case, liver tumors if there is a recurrence. So these are all options and can be quite effective in treating fibrolamellar in the right circumstance. When we think about systemic therapy or drug therapies, and I am a medical oncologist, so this is something that I do a fair bit of, we consider it in two major circumstances. One is if there are multiple sites of disease where we don't think we can safely treat them with these other local therapies, or in some cases we think about it in patients who just keep having spots popping back up at increased frequency. And we have to think about, the pros and cons of our treatments. We think about, what is the experience in terms of, what has been seen in the past with different drug therapies in terms of the likelihood of shrinking or controlling the cancer or the efficacy. Patient preference, how aggressive patients want to be and what side effects they would be willing to tolerate. And different treatments have different side effects. So we often will discuss the pros and cons of each option and make a decision jointly with our patients. And then there are some considerations involving logistics where that treatment should occur. If someone's coming from the middle of the country, sometimes I may coordinate with a local oncologist and just guide the decision making, but maybe some of that treatment may be done closer to home if that's the preference of the patient. In terms of the efficacy of treatment, I think it's important to understand that this is a very rare tumor, and we are continuing to gather information over time. And one thing I'll highlight is that we are currently involved in a collaboration with multiple institutions trying to pool all of our experience in fibrolamellar and quantify what have been the outcomes with each specific drug therapy across the United States. And this is, really an important area and important to get more information, just pool all of our experience. And that is ongoing for sure. Clinical trials are certainly one option to consider, and I do get involved in clinical trials. The options for what clinical trials we might use are going to depend upon our increased understanding of fibrolamellar. And we have trials that are available either across the country or here at Seattle Cancer Care Alliance and University of Washington. Some of these are phase 1 trials, which are relatively early experimental interventions with newer drugs. And we try to select those trials that would apply specifically to our understanding of fibrolamellar. So if trials are available, if a patient is looking for a treatment, I look into our portfolio of trials, and we usually have many clinical trials, some of which could apply to fibrolamellar. And I bring that up along with the more typical chemo therapy options and discuss the pros and cons of each with my patients. I do sometimes refer to other centers. This is a rare disease, and trials are often available at other sites. And if they apply, I will bring that up. So I try to stay aware of all the different options across the country. And we sometimes get access to drugs through other mechanisms other than standard clinical trials, but those can apply as well. Next slide. So this is just, I went through and tallied some of the phase 1 clinical trials just to highlight that we do use clinical trials, and I think they can be very appropriate for patients with fibrolamellar in the right circumstance. It's not always the right treatment, but I'm always looking for trials that would make sense. And so we've treated patients with immunotherapy type drugs, trying to enhance the immune response to treat fibrolamellar. We have used chemotherapy plus other agents to try to augment chemotherapy. And as you can see, we have had a mixed result, and this is still certainly an evolving topic. But I think there are, as we get more understanding from the research side, we get more options that are make more sense for our patients with fibrolamellar. And I, as I mentioned, do send patients to other sites. So I've sent 2 patients to UC San Francisco when there was a trial specific for fibrolamellar. And I also, as I mentioned, access drugs through compassionate use mechanisms if I think it makes sense and after discussion with my patients. So next slide. So as I alluded to, the clinical trials, as we get more data, we have different options. And our trials will often change. So every 6 months or so, we will have a whole new set of clinical trials opening up. And I normally try to guide which trials we open up, in part based upon whether they look appealing for patients with fibrolamellar. So we have a new protocol that is now just opening up, which may get to starting to look at some of the research that Dr. Scott and Sophia mentioned in some of the later talks. So I look for trials that make sense specifically based upon our understanding of fibrolamellar. At this point, there are actually 2 clinical trials, one that opened since I put these slides together. So one is at Johns Hopkins, and I collaborate with my colleagues there, and have discussed sending some patients for clinical trials of a vaccine against the mutation involved in fibrolamellar in conjunction with immunotherapy. And MD Anderson has just opened a trial of chemo plus immunotherapy-based drugs, where we would certainly consider sending patients there as well. And then in our phase 1 clinical trial group, we currently have 31 clinical trials of experimental agents. And some of them would allow patients with fibrolamellar, and in the right context, I would consider that. Although, as I mentioned, these trials continuously change, and we continuously reassess whether they make sense for our patients. So with that, I will hand our slide share over to Ray Yeung, who's going to talk a little bit about the bench to bedside to bench transition and the importance of that.
Thanks, Bill. Hope you can hear me. I will talk a little bit how we got into this. So before we start, I'm a surgeon scientist at the University of Washington. My primary focus is related to liver cancer of all types, primary and secondary. And over the last 20 years or so, our group has taken care of approximately 24 of these fibrolamellar patients. And we continue to try to do our best, as Bill and Kim mentioned, to serve this community. So in the next couple of minutes, I'm just going to give you a very brief, sort of a historic perspective as to why we got into this. Okay, as you heard, most of these patients have absolutely no history of liver disease. This is just a random event, if you will. Most oftentimes, they come to us because they have persistent abdominal discomfort, and they're usually brushed off at the very beginning, saying, oh, you're just playing sports and you hurt your muscles or whatever. But the persistence of the symptoms usually leads to some sort of imaging. My point is that by the time they come and see us, it is usually pretty advanced, as shown in this situation where you can see a big mass occupying the left side of the liver. Fortunately for this particular patient, we were able to remove it safely while still relatively early in the follow-up period. The patient remains disease-free at the 3-year mark. I just want to recapitulate what Dr. Riehle had said. The beauty of liver surgery is that in an otherwise normal liver, the remaining part will compensate. But I want to dismiss sort of a misconception that when we talk about regeneration, when we take out the left liver grows back. That is not the case, as you can see here. The left liver is absent for life. The only difference is that the remaining right liver takes over and usually gets bigger or hypertrophies over time. Unlike that patient, not infrequently, we have situations where, like Dr. Harris says, after the initial resection, the disease comes back. And in this particular fellow, after his initial resection, he was doing fine for a few years, and then it recurred, usually within the abdominal cavity. So we had to go back. And the reason is I treated this fellow over 10 years ago now. And back then, even fewer therapeutic options. So the surgery was the only really potentially curative option at that time. And every time it recurs, we go back. But the interval of that recurrence becomes shorter and shorter. And as you can see by the picture, he even tattooed himself every time he comes in and say, well, just do what you have to. Unfortunately for him, after multiple recurrence, it became so widespread that there was no other therapeutic option. And it was at that point that I sort of promised his mother that we would really study this disease and try to find better systemic option for this rare cancer. So for those of you who haven't seen it, this is what the tumor looks like when you cut it open. It doesn't look very appealing. But the point is that when you look through different parts of these tumors, they look different. It's very heterogeneous, if you will. So, but if we take a closer look under the microscope, it becomes even more complex. On the left side of the screen here is the normal liver as compared to the right side, which is the tumor. So the question to us is, how does a normal liver like that become cancerous? And as you've heard already, the main change that happened in this disease was the discovery of that mutation by Senny Simon at Rockefeller. And what they found was that there's a common mutation that results in a fusion protein, now known as DNA-J, PKC. When I first read the paper, I knew nothing about this, so I looked it up. And lo and behold, I realized a ton of the work that has been done in the field of the protein kinase A was done right next to me in John Scott's lab, just one floor down from my lab. So I rushed over to John and said, hey, you know, there is this disease. Do you know anything about this? And that's when everything started, right? So he and his group is going to bring you up to date on some of the very exciting work that they're doing. But in addition to that, you can see there are a lot more things than just the tumor cells. Here's a different highlight showing you the non-tumor component, the non-tumor cell component of the tumor itself. And the brown cells highlight the macrophages, here using a CD68 staining. But they are in partnership with a whole bunch of those small round blue cells, which are lymphocytes or T cells. So what are they doing there? I'm sure they're not there by coincidence. And you just heard Dr. Harris say, well, we're beginning to study this in terms of the immunotherapeutic implications. So again, fortunately, my partner in crime, Dr. Pillarisetty, who is also a surgeon, has a strong program in tumor immunobiology. So we got him on board to also study this from that perspective. So I'm going to hand it over now to John to introduce the research team at the University of Washington.
Thank you, Ray. And yeah, I would just echo what Ray said, that we spend a lot of time running up and down stairs. Our labs are really close together. And I think that that's a really hugely important aspect of this, that we are able to work so closely together. So I'm going to sort of pick the story up from where Drs. Riehle, Harris, and Yeung have left it off. And it really begins with this mutation. So we're now really we've gone from the liver to the hepatocytes, the liver cells. We're now going to the molecules. We're getting much smaller. So as Ray correctly mentioned, there is a mutation that was identified in 2014 that results in the formation of an unusual enzyme called DNA-JPKAC. The important thing here is the blue part is the normal part, we think, and the orange part is the part added on. What happens in FLC patients is they have in their tumors, they have two copies of this enzyme, one abnormal one and one normal one. And so it's the abnormal enzyme that causes the problems. So where we began to focus on this was actually on the orange part, because that was the unusual part, and discovered that that segment serves a bit like some sort of molecular Velcro to recruit additional enzymes that are known to be involved in cancer to create what we call a signaling island. And it's really studying those complexes that are somewhat unique in most fibrolamellar patients that has driven our research and got us towards clinical trials. So here is a normal liver cell, and the only thing to focus on are the yellow dots. There's not very many of them, and those represent those signaling islands. So this cell's perfectly fine. Here's a modified cell with the gene for fibrolamellar. A very different picture, many, many more dots. Much more importantly, again, following on a different level of resolution from what Ray showed, here's a section of normal liver and FLC tumor where you can see the same thing. So we knew that these signaling islands, which are sort of groups of enzymes, are formed in fibrolamellar, and this would be what would be considered the molecular target for our research, with the hope to develop or identify drugs that could selectively target parts of this complex and inactivate it or make it ineffective. So it was at this point, once we'd identified this, that we could actually initiate a drug screen. And this originally was Ray's idea. And what we decided to do was to do a drug screen of known cancer agents that had already been FDA approved. Now, the advantage of that is that these compounds, there's a lot known about them, and it bypasses much of the really important legislation that's necessary before you can be given to patients, because these are approved for patients for different contradictions, for different diseases. So we wanted to just see if we could find any of these compounds that might be repurposed for FLC. And it's a very complicated process that's way beyond my abilities to understand. It involves artificial intelligence, bioinformatics, and a lot of computers. But the simple way I understand it is that you take your cells, you give them different combinations of these drugs. There's 125 drugs. If the cells grow more rapidly, they're resistant to drugs, so they end up in the top left-hand quadrant. If they're sensitive to drug, they exist in the bottom quadrant, so either the turquoise or the green quadrants. So here's normal liver cells, and as you would expect, these compounds have little or no effect. Here's what happens when we look at cells that express the FLC protein. A very different pattern. Many of these drug combinations or drugs enhance growth, so you don't want to use those. But these three in the bottom here significantly retard cell growth. And what was absolutely astonishing to us, and to me in particular, was that these three compounds all target the same enzyme. They target this enzyme called MEK. Now, we've not really talked about MEK much, but when Dr. Harris talked, he mentioned another enzyme, which you'll see in this signaling island called ERK, which communicates with MEK. So if you block MEK, you block ERK. So we immediately thought that this would be the molecular target. And so we made this observation and could now begin to move from the sort of cell biology and biochemistry towards the pharmacology, because we had combinations of drugs that could target different elements of the signaling island. And you can see that when you use these drugs together, which is the green line, you are able to kill the cells. Now, there's one caveat here, and a very important caveat. That's killing cells in a test tube. And so we wanted to take it further than that. And this is a really important point I want to make. We're excited about the progress we're making, but we realize that what's done in a test tube or done in a cell line can be a little bit different inside a tumor. And that's why we have to take this complete holistic approach. So what you're looking at here, the only important thing is to look at the top black band. That is the enzyme ERK that we talked about before, that there's more of it in rapidly dividing cancer cells. You can actually see that if you stain the cells. You can see it below here. And when you have the mutation for FLC, you get more of this molecule. But this is where the conundrum comes more complicated. We then repeated all of these analyses in patient samples that were provided by Drs. Riehle and Yeung. And you can see that we got a very different pattern. In fact, there was no pattern. And it took us several months of scratching our heads to try and figure out what was going on. And then we realized something, and it relates to what's already been said about the kind of heterogeneous nature of FLC. It's a tumor with lots of different kinds of cells in different states. And so if you look at this image here, the only important thing to look at is the yellow image. And you can see that there's different degrees of staining. And this was a picture that was taken in that microscope I showed you earlier. What that actually means is that there are different levels of this enzyme and its activity is different. And this is the enzyme ERK. We can look at an enzyme that communicates with ERK and we see the same thing. What this means is that if we are using a drug therapy towards this group of molecules, we have to use a significantly high enough concentration of the drug to be able to work at the cells with the highest level of the drug in it. So that's a really important part as we begin to think about using these compounds for the first stages towards a clinical trial. Here's another way of looking at this. This is an assay that looks at cell growth. You can see that in the control, there's a lot of purple cells. And when you use these drug combinations, you can pretty much kill the cells, which is encouraging. And it encourages us to think more about moving forward into animal models and into patients. We've done this with a variety of these compounds that we identified, and we're fairly confident of this result. So by way of giving you more of an idea of what we're doing now, and we're absolutely at the kind of frontier of where our knowledge base is, we've also been able to take advantage of genetics to be able to disrupt this complex, to know a lot about its mechanics. We can remove the molecule that holds everything together, the cells die. That tells us this combination is important. And very importantly, we can remove the mutant enzyme and the cells die. And so this is really where I'm going to stop telling the story, and it really sets the stage for Sophia Lauer, who is a PhD student in our group, to tell you about this really exciting research that she's conducting that builds onto this and really explores much more in detail about what is going on under these situations. So, Sophia, I will pass this over to you.
Thanks, John. Go ahead and share my screen. Oops. Okay. All right, so hi everyone, my name is Sophia Lauer and I'm a PhD student in the Scott Lab here at UW Pharmacology. I'm really excited to talk to you today about some of the FLC research that I've been working on. So as John mentioned, the focus of my project is really determining what's happening at the molecular level that's causing FLC. And if you remember from his presentation, he was talking about this unique protein that we think might be a major player in the development of FLC. And so to show you kind of how things look in healthy cells, you have this protein called pKa, which is a type of enzyme called a kinase. And in normal people, you see only this turquoise part. But through a genetic hiccup in FLC patients, you end up getting this extra orange bit that's called DNAJ, and that's fused to the turquoise bit. And so you end up with this fusion protein, DNAJ PKC, that as a whole is associating with these signaling islands that John mentioned. Our lab has recently discovered that the extra orange bit that's added on acts like a piece of Velcro that brings in other proteins, such as this red molecule here, that's called HSP70. And this is known to be upregulated along with kinases like PKA in a lot of cancers. And we know that having HSP70 next to a kinase, like in this diagram, is a common theme in FLC. So it's possible that this red molecule, HSP70's interaction with the turquoise molecule, PKAC, is also responsible for driving this cancer. And so the other part that John talked about is that these enzymes are brought into these signaling islands. And in FLC, these islands recruit these additional proteins, shown here, which are upregulated in around 30% of cancers. So really, if you add all of these things together, you end up with all of these molecules in one place, in the wrong place at the wrong time. And that combination pretty much gives you FLC. So as an extension of these recent findings, for my project, we hypothesized that this DNAJ-PKAC fusion protein is responsible for driving FLC. And more specifically, we want to identify which part of the fusion enzyme causes FLC, because as you remember, the fusion enzyme is made up of these two parts, so the turquoise bit and the orange bit, and they each do different things. So what we're trying to determine is if it's the activity of this turquoise portion, the kinase, since it's known that many kinases have the potential to cause cancer, or it's this orange portion called DNAJ that brings in other oncogenic proteins. And so with these hypotheses in mind, the next logical question to ask is really, how can we test these? And what's exciting about research is that if we have an idea about what protein is responsible for driving the cancer, we can try to artificially create cancer's liver cells by putting that protein into normal liver cells. And so what we decided to do was use a relatively new field called chemical genetics inside liver cells to form different combinations of proteins. And by stitching together these molecules using drugs, we aim to essentially recreate the molecular features of FLC in these liver cells. And so here's a compound that's basically going to be what's acting as the molecular glue that we're going to use for this chemical genetics system. So we started out with the two parts of the fusion enzyme, the turquoise and the orange portions. And then we're going to add these sort of molecular handles to these two halves that will allow us to artificially induce the formation of the DNAJ-PKAC fusion enzyme that's found in FLC upon addition of this compound here. And if doing so in liver cells causes the cancer phenotype, then we know that this enzyme could potentially be the thing that's causing FLC. Additionally, to test our second hypothesis, we're going to bypass the orange bit altogether and have HSP70 interact directly with PKAC using our molecular glue. And if we get the same effect, that would tell us that the orange part of the enzyme is what's important, because as I mentioned earlier, the orange bit acts like a strip of Velcro that's going to bring this red molecule to the turquoise one. And so this is essentially what I'm working toward with my experiments. I'm using pharmacology inside normal liver cells to either induce formation of this FLC fusion protein, DNAJ-PKAC, or mimic the HSP70-PKAC interaction in order to produce a cancerous phenotype. And so far, from a biochemical standpoint, the system seems to be working. So here's an experiment where I created these two synthetic genes that I could put into liver cells to get them to produce each half of the DNAJ-PKAC fusion protein. And then, as I described in previous diagrams, I added a chemical compound, or the molecular glue, to the cells that would cause the two halves to basically come together and form the fusion protein. And as you can see in lane 2, DNAJ is associating with PKAC in the presence of this compound, but not in the absence, as shown in lane 1. So this essentially tells me that formation of this fusion protein here can be induced in the model system that I'm using. And so to help you better visualize how this inducible fusion process looks within these cells, here's a video showing live cell fluorescence microscopy of the fusion complex being formed. And you can see here that there is movement of the diffuse yellow signal to the membrane of the cell. And this is essentially showing us that we're forming the cancerous enzyme complex. And the takeaway from this video is that we can artificially induce formation of these FLC complexes. And so the next thing I wanted to do was see the effect of forming these complexes on cell proliferation. And so here's an observation of p-ERK or phospho-ERK signal, which is something that can be used as an index of tumor cell growth. And the important thing to look at here is that in lanes 2, 4, and 6, there's a darker signal indicating that these cells are dividing more. This means that when we induce formation of this HSP70 PKC interaction, it causes the cells to increase growth. And so this points to a potentially important role for HSP70 in the pathology of FLC. And more importantly, there are drugs out there that can block these enzymes, which is something we're going to be looking into. And so in sum, we've gotten to this point where we've created a simplified system that allows us to answer some of the mechanistic questions about what's going on at the molecular level in FLC. But this is really just the beginning. Dissecting the system biochemically in liver cells allows us to definitively test how different kinds of drugs would work in each of these situations. And here are a couple of the combinations we're going to be looking at. But our ultimate hope here is that we can use what we're finding to help us reverse the disease in human tumors. And we're really excited to be working towards that. So I hope this was helpful in giving you a little insight into what I'm doing. And now I'm going to pass off to Alan, who will be discussing how we use patient tissue samples to further our research and understanding of FLC.
Great, thanks, Sophia. So my name is Alan Utria. I'm a pediatric surgery research fellow working in the Yeung Riehle lab. And I'll be talking to you guys today about the tumor slice model that we use for studying fibrolamellar carcinoma. First off, I'd like to thank the FCI Foundation for organizing today, as well as for giving us some funding in the past, as well as the Department of Defense, who we're currently funded by. And most importantly, I'd like to thank patients who have donated tissue to our lab and who are the ones who make this kind of research possible. So the main benefits of this model is that it allows us to take a tumor and, while keeping it alive, divide it into many similarly sized pieces, preserving the original spatial architecture and cellular diversity of the tumor. And so then we can then study how the tumor cells respond to different treatments. And in many ways, this is the closest we can get to try mimic how a tumor may respond in a person. So the first step for us is on the day of surgery, for patients who have consented to donate some tissue to the lab, the surgeon will call our lab and we'll go to the operating room once the tumor has been removed. And on a back table away from the patient, but still within a sterile setting, we use a core needle, core punch biopsy demonstrated here to essentially remove small cylinders of tissue that we can then place in fluid to keep it alive and on ice. Once we have that, we go to our lab and we use this machine that's called a vibrotome. And it's essentially just a very expensive, fancy slicer. And we take our cylindrical tissue that we remove from the operating room and we cut it into very thin and consistent slices of tissue. We can then place each slice in an individual well on a plate, which allows us to basically run a whole variety of experiments, treating each slice as its own mini experiment within a bigger experiment. So the different things we can do with these slices is various types of treatments, which include immunotherapy treatments, where we might modify immune cells, such as macrophages or T cells, to try target the tumor, or we can use antibodies that target the tumor, as well as a variety of drugs that have already been FDA approved, or drugs that are in clinical trials. We can treat the slices like this for sometimes up to even 7 days before we assess the response. When we go to assess the response, there's also lots of different things we can do. So we can look at the slices under the microscope, just do basic tissue histology and see how things are changing. Or, as already demonstrated, we can do live microscopy and see how the cells are changing or interacting with each other. We can also look at what populations of cells are present within the slices and how those populations might change under different treatments. We look at protein expression and gene expression, which can give us more insight into how these tumor cells are different from the non-tumor cells, and also how these tumor cells might change under different treatments. And ultimately, we can look at the tissue viability and see what kind of treatments are causing the tumor cells to die versus which treatments have no effect. So as you can see, this is quite a versatile model that allows us to use patient tissue to learn a lot about the disease and move forward towards developing new therapies. And with that, I'm going to pass it on to Dr. Pillarisetty, who's going to talk more about the immune aspect. Thanks.
Thank you, Alan. Let me just share my screen. Okay, is that full screen? Great. Well, thanks very much. And, you know, I've also been extremely lucky to be part of this group. And as Ray mentioned a few minutes ago, you know, his, you know, clinical experience and work with these patients really drove a lot of our group kind of moving towards taking care of or trying to understand fibrolamellar carcinoma. And I'm definitely part of that initiative that he led. I've been interested in liver immunology for almost 20 years, since I started working in a research lab when I was a resident, and found that that's a really fascinating environment, and that the liver is a place that is designed to you know, sort of accept things that are coming into the body. Because if you think about it, everything we eat ultimately winds up getting processed by the liver. And, you know, you don't want to have allergies to every single thing you eat. So the body needs to learn how to, you know, accept the stuff that goes into the liver. So I want to set the stage here a little bit, just talking about the way that you can try to understand the tumor microenvironment, and especially how that relates to immune response. So what I happen to be showing here is a pancreatic cancer. Doesn't really matter much except for the concepts that are going to come about in this. And what you're seeing is a lot of different cell types. And this was mentioned earlier. And I've got kind of like little mini fake cells that are in here as schematics. And what you're trying to see is, like, here's cancer cells, here's fibroblasts, there's some T cells and macrophages. And if we start thinking about each of those components separately, we can think about how we would target each one, or how they interact, and how that would alter, you know, ultimately killing the cancer cells. So, you know, my world of immunotherapy, the first thing that people think about is T cells. And typically what you try to do, and Alan kind of mentioned this, is, you know, one option is just add more T cells. You can add them. The other is you can activate them. And you can't open a magazine today without finding ads for drugs that activate T cells. But they don't always work. But we've seen sometimes that it does work even in fibrolamellar cancer. Macrophages, as Ray Yeung mentioned earlier when he showed one of the images, those are the cells that process proteins, like things, you know, if they come into contact with something that's abnormal, they might chomp up that cell, hence the name macrophage, they eat. And then they have to decide, what are they going to do with that information, right? Are they going to tell the body, oh, and tell the T cells, don't worry about it, this is something that's okay? Or are they going to say, okay, activate and start killing the tumor? So for those, we can either remove them, or we can try to turn them more into activated macrophages that are going to lead to a tumor killing activity. Stromal cells, or fibroblasts, these are the cells that that are basically like scar tissue. And in a lot of ways, cancers are like scars, except that they're scars growing out of control that can spread. There's a lot that the fibroblasts, we're learning more and more, are doing to affect how immune cells function. And some things that we can do is alter the molecules called chemokines that attract other cells, and then ultimately free those other cells. And then ultimately, you know, the big thing is to try to kill cancer cells. So that's what John Scott and his group have been talking about, obviously, is drugs that can directly activate you know, pathways that will lead to the death of these cancer cells, or lead to their susceptibility to drugs. So fibrolamellar carcinoma also has a complex tumor microenvironment. What you're seeing here is a multiplexed immunohistochemistry. What that just means is there's lots of different antibodies staining different cell types, and different colors represent the different cells. And fundamentally, you're seeing T cells, you're seeing macrophages that are stained in there, as well as activated immune cells in general. And if we look at what we found, if you look at a fibrolamellar carcinoma, about half of the cells, at least in our study, were the cancer cells. There was a whole bunch of what we called other cells. Probably more of those were stromal cells, which we didn't stain for. Macrophages were a small group, and T cells were another small group. And you saw in Alan's previous slide, this particular image. This is from a collaboration with Kevin Barry, who is now at the Fred Hutchinson Cancer Research Center, but used to be at UCSF, where they took apart a tumor and broke it up into single cells and then did genetic analysis of all the cells and showed how specific different groups of T cells existed within that tumor. So fundamentally what we figured out, and we have a paper that we just submitted on this, is that it's a very immunosuppressive microenvironment, even though there are cells that theoretically could kill the tumor. So we use the slice culture model that Alan was mentioning to try to kill the tumor cells using different drugs. What you can see here is comparison of just a control group and then a drug that helps move T cells around. Here's a PD-1 drug that just causes T cells to be activated. And then here's a combination of the activating drug and the one that helps the T cells move. And then here is something that blocks a cytokine that is known to reduce T cell activity. So when we use these different treatments, you can sort of tell when you look here on the images, but on this graph, what you can see is more clearly staining for cells that are dying. And with the control, very few cells are dying. With the combination, with the thing that makes T cells move, not much. With a thing that activates T cells, it goes up. And also when you combine with the drug that moves T cells and activates it, maybe even more. And then also when we block that immunosuppressive cytokine signaling, you can see activation that's leading to cancer cells death. This is a zoom up to really just see, this is what the control tumor looks like at 6 days, and it's really starting to get beaten up when you use that other drug. So the point here is that it looks like when we get fresh tissue that we can actually make some of these cancer cells die with immunotherapy, and I think we're at the beginning of some exciting stuff. So thank you.
So thank you, Venu, and thanks everybody for listening to this kind of sojourn from the bed to the bench side and moving back. So at this point, we're going to transition, and David and Mark Firth are going to be leading a panel discussion. And I'd like to encourage you to send questions through the question and answer feature on Zoom so that we can try and effectively answer the questions as best we can. And with that, I will pass this back to David and Mark. Thank you.
Thanks, John. Like John said, if you have any questions, go ahead and click on the Q&A that's on the bottom of the bar, and just submit your questions there. The first question I have is actually more geared towards Bill Harris. How would you, let's say you live in a place that does not have a big hospital like UW or even like MC Anderson, how would you go from going to a smaller clinic to then being able to get to a bigger clinic like UW or those type of places?
Yes, so I guess I can speak to that a little bit about what historically has been done, and then now that things have changed in part due to COVID. So, historically, what's happened is patients are often, diagnosed with a liver mass, of some form, and often it's perhaps misdiagnosed out in the community. And, I think anyone with an atypical situation where they have a liver cancer that is unexpected, or a liver mass in a young patient, we think really should be evaluated by a larger center. And we tend to have referrals, nationwide, but more specifically, I think we get referrals from about 5 states to University of Washington and our liver tumor group, where Ray and Venu and others are certainly involved, as well as myself. Kim is involved at Children's Hospital as well. And, I would say that anytime you have an unusual situation, getting to a larger center makes a lot of sense, and getting a multidisciplinary evaluation. And often we make the diagnosis in second opinion when we're seeing these folks. So I think if the problem is that, many of the folks on this call, already that diagnosis has been made. but it's the other folks who are out in the community where maybe it's called more typical liver cancer. If it's an atypical situation, getting a referral to University of Washington or Children's Hospital is critical. And even if you have a diagnosis of fibrolamellar, many folks aren't as familiar with that. And I think getting to a larger institution that has some experience with this, at a minimum for a second opinion, is critical. So I'd say reaching out to a major center that has experience with liver tumors in general and fibrolamellar, if that's suspected, is key. In the COVID era, we have been able to do a lot more telemedicine visits. So, now I can actually, and we as a liver tumor group can do telemedicine visits and provide consultations from afar. And we're licensed in multiple states to do so. This is because, patients can't travel because of COVID as easily. But I think that actually opens up some options if you can't travel directly for an evaluation, to at least get a complete assessment via telemedicine. And I think that's a very nice option. And sometimes I see patients who are out in, Montana or Wyoming or Alaska, where maybe they can't travel for treatment. Maybe they just need surveillance and for us to track it from afar and, keep an eye on it, and come out when it's more interventions are needed. But sometimes we can coordinate with a local oncologist or other group just to provide input on what kind of surveillance would be required, things like that.
Well, thank you. My next question is kind of open-ended to anyone who wants to answer. What was kind of the driving factor that wanted you to start researching and working on fibrolamellar?
I think there's going to be a lot of different answers to that. Maybe I'll start. In my case, as Ray said, I've worked on this particular enzyme family for a very long time. It was a conversation with Ray, and I think that's important for the audience to realize that research and clinical research in particular is a fairly isolated process. And what's really fabulous is when you can work with very talented people who also become your friends. And I think that's a big part of this, that we're also located fairly close together. Ray, as I said, Ray and I, literally, he's upstairs from where my department is. And that's a huge advantage. I think what Bill said as well is really important. We have evolved over the last 11 months to having conferences like this, and it really opens up the networks. I know that I've set up meetings that Sophia and I have been part of that go over 10 time zones, where we just have weekly group meetings over 10 time zones. And that really speeds up the passage of information. And then I think a third part that is really important, that is, I really want to emphasize how much this has been brought on by the Fibrolamellar Foundation, is the idea of openness and sharing information. And that's something that we do as a group, but we do it with other groups around the country. And, Mark Firth and John Hopper and the Davises have really led that. And that's a hugely important part, that we're not keeping secrets from each other. We're all working together because we recognize that, we need to be doing this as fast as possible.
I just want to echo that, in that I think the FCF serves a very important role also in disseminating knowledge. Right? This is such a rare disease. They are a pivotal point where patients, once diagnosed, can go to and ask all those questions that you have raised. And they can help patients navigate and help them get the proper care at specialized centers such as ours throughout the country. And I think that's really, really beneficial.
Yeah, and I'll just say that, one of the reasons I went into the field I'm in is because liver cancers in general, there are many different rare forms of liver cancer. And then liver cancer in general is a complicated area to treat, where it involves interacting with a lot of colleagues from different specialties. When I went into this field in general, I felt like there was a lot of need to really further research and better our treatments for all of our patients, including patients with a lot of different rare forms of tumors like fibrolamellar. But what really got me into fibrolamellar, more heavily was my patients. So I started seeing more patients with fibrolamellar, and they actually recruited me into the Fibrolamellar Foundation and got me involved. And then I think the Fibrolamellar Foundation has really been furthering research in a great way. And I think they've been recognized by many as doing an amazing job at sort of improving how we research rare tumors. So I would applaud that. But in the end, I did it because there's a need in the end. And I think, thankfully, there are some promising leads for research that we can move on here.
Hey, gang, there's some good Q&A right now that some of the folks have put in here. One of them is asking about markers for FLC. And, what's the status of that? So the question basically is, anyone working on finding markers for FLC, which if tested for a young person would get that FLC patient into treatment early enough to successfully resect before metastasis? Anybody want to field that one?
So I can, this is, it's a tricky question. I'll just say that the first part is just making the diagnosis in general. And I think that, we have pathologists who have been involved in confirming the diagnosis of FLC. We have the genetic changes to identify FLC, which are critical. The next step is trying to find markers to predict who is at higher risk of recurrence of FLC. And that is an area of uncertainty. What I can tell you is that there are some leads there that we're starting to think about. And we have sent blood samples off to try to test this in conjunction with some other researchers over the years. But right now, in other cancer types, they're starting to be able to identify circulating DNA from a tumor that seems to be a good marker to predict the likelihood of recurrence of a cancer. And these tests are quite new. They evolve over time, but there's a new type of test just becoming available. And I am in discussion with this company about whether they can do this for patients who undergo resection for FLC to try to figure out, do they have microscopic disease left over? And I think that is something that we're starting to look into as to whether we can start collecting DNA from the blood to see if that might be helpful. And those discussions are sort of just underway, but I think it's an important area to pursue.
Thanks.
And I'm
Bill.
open to other comments on that as well.
Yeah. Thanks, Bill. I was going to say, gang, it's 1:30, and if people want to stay on for another 10 minutes, I think this is important enough. If people who are attendees bow out, we will again have this recorded, and please, you can ask questions via the email. So again, if people want to stay on, which I hope they will, David or Mark, I'm going to pass it to you to field some of the other questions in the queue here.
Thank you, John. There are a number of great questions. I'll try to get through a couple of them in the time we have. First, a general question for anyone on the panel, particularly I think probably John and Ray, is how long do you think it takes to go from the bench back to the bedside in the process you've been talking about? And what are some of the biggest hurdles to take an idea like a new combination therapy to patients?
I think that's a really important question. I think the first thing, we're excited about being able to do that, but the important thing is we want to be really realistic and honest to make sure that people realize that when you're doing trials such as these, it takes a long time, because as I tried to demonstrate, we can do one thing in an isolated cell, it's very different in a tumor, particularly in this case. And so while we're very excited and optimistic about the direction we're going, it's very hard to put a precise time on when this can all be done. What would you say, Ray?
Oh, I totally agree. I think the important thing is what we find in the lab could be very exciting, but how translatable that is to the bedside is another question. There are lots of hurdles, especially in this case. In my previous lifetime, I was involved in another rare disease called LAM. And fortunately, there was a therapeutic target. Even then, it took almost 10 years to go from bench to FDA approval. So, but we're optimistic.
Thanks for that to both of you.
I think another important point is that we think we've identified the molecular target. It's got two parts. If we're really honest, we still don't know if one of these parts predominates over the other, and whether it's the orange versus the blue, or they're both involved, or it's the recruitment to a signaling island. So we've made tremendous progress, but it's like anything, unfortunately, when you discover something, you only open more questions to be answered.
We have a couple of questions that are at kind of a more molecular level, and I'm not sure who would like to carry this one, but one is the cause of the chromosome 19 deletion understood? And is that deletion continuing to be found in all fibrolamellar cancers? Or is there a little more diversity than that? John, you want to take a crack at that?
So, yeah, I'm looking around at my colleagues and I'm seeing some smiles. Correct me if I'm wrong, but it's in at least 80 to 90% of the patients. There are a few other fusions that are found that actually relate to other cell signaling molecules, but this is by far the most common one. And I think that while it's important to focus on this, we have to be open to the possibility that there are others. But again, it's this idea of whether this mutation is the cause or it's a consequence. And those are two different things, and we want to design research and projects towards addressing those. And that's a little bit of what Sophia's trying to do. I would pass that on to Kim and Ray and Bill.
John, maybe I'll suggest folks can write to the foundation about that, and I happen to have a recent manuscript that a number of us are working on that answers those questions. So be glad to share that with people. But fundamentally, it's a very large majority. The other, another question I wanted to raise that actually reflects a couple of different questions that have come in is specifically for patients who want to share information about their cancers and also about their ability to donate tumor tissue and how important that is to research in fibrolamellar. We've already seen a couple of examples from you of use of tumor tissue. There are a couple of possibilities that I think many of you are aware of. There's a registry that's led by a group from Rockefeller University, the Simon family and Sandy Simon. There is a natural history study that's opened up at the National Cancer Institute, which is recruiting fibrolamellar cancer patients to provide information about their history and also specimens to get, when possible, to get genetic information. What other kinds of, and there's also a biobank for fibrolamellar tissue, which we are leading as a foundation that's based at Mass General, but which is distributing tumor tissue to any lab that applies for it, essentially. And I think there's also a specific collection of tumor tissue at Rockefeller. So can you guys add to the importance of having access to specimens and to patient data? And What should patients think about when those questions come up? Bill, maybe you've come into that as something your patients have asked you about.
Yes. So I would say that, as you alluded to, there are multiple efforts, trying to collect tissue, and I think the FCF has been facilitating that, and I've been in conversations with the FCF trying to get samples to you. We also have, we try to collect tissue to facilitate research, and that's predominantly driven through one mechanism, which is Ray Yeung has a biobank here where we try to collect specimens. I may defer to Ray to talk about that, but it's critically important to get tissue to further research, and we are very open to distributing samples. We will, if we get tissue for research, we also want to try to distribute those samples out to as many other people as possible. And the FCF has been helping guide that as well. But Ray, any comments? Because you run the biorepository here.
Yeah, so we do have long-standing biorepository here to collect. So any patients that we operate on, we collect the tissue. I just want to make one point. I think that is probably the most important contribution that the patient feels that they can do in this field, and that is to donate the tissue for the purpose of research. And for rare disease like FCF, we are so much in need of that to create model systems, which we desperately need in order to study. So we try to share any of our samples with the FCF Foundation and the community as a whole.
Yeah, thanks, Ray. And
yeah,
every specimen that comes to the biobank in Boston immediately goes to the Broad Institute across the street for model creation, to a laboratory that's putting tumors into mice for model creation. There are immune cells going to a lab at St. Jude, and sometimes back to you guys. So it doesn't sit in the freezer. It actually gets used.
And I'll just highlight one other point on this, is that the types of samples we get in part depend upon planning. So if someone has an upcoming planned surgery to remove a tumor, that is a situation where if we can sort of get everyone involved early and plan to collect that tissue in a good fashion, we can get a lot of important information out of those samples. And I would say, if there's a planned surgery and time to plan for it, getting the FCF alerted, if it's here, getting us alerted is critical.
Yeah, we just had a question about whether collection for the biobank was possible from outside the United States, specifically the UK. We don't have a mechanism for that at the moment, but we'll work on that. So please follow up with us. That's a very important question. I'd like to come back to
I would do one or two more questions at the most, and then we'll close it down. I know we have a lot on here.
Thanks. So let me come back to a more medical question, which relates to a point made very early, which is that the diagnosis of fibrolamellar is often delayed, partly because otherwise these are healthy patients, maybe also because there isn't enough knowledge about this particular cancer, which we heard about from David from his personal experience, and hasn't changed as much as we might like. Are there things that can be done to improve that situation? For example, finding specific markers that might allow a doctor to diagnose fibrolamellar even when symptoms remain fairly vague? Bill, you want to? I see your face on that.
It's a tricky question, because many of these folks will not be coming to a larger center with more knowledge of fibrolamellar early on, and so a lot of this has to do with the need to enhance education to the people who would be involved in the early diagnosis of fibrolamellar. I think that's increasingly happening through FCF and education, as well as just as more research is done and more information is discussed in conferences, medical oncologists and gastroenterologists and others are increasingly aware that fibrolamellar exists. Educating radiologists is important to let them know, as Kim mentioned, there are certain imaging findings more specific to fibrolamellar that can lead you down that road. And then confirming with things like the genetic testing that I mentioned, that's increasingly available as well. But it's difficult because we have to really reach out to lots of different physicians to increase their awareness. And I think awareness that it exists is probably the #1 barrier right now.
Thanks for that, Bill. Kim, did you want to add something?
I was just going to say that I think in pediatrics, people are maybe a little bit more accustomed to reaching out about rare things. And we have a structure nationally called the Children's Oncology Group. I'm on the liver tumor committee for that group. And so not infrequently, community pediatricians, if they find a liver tumor like this, will reach out to that committee to kind of get advice about next steps. I think one of the questions was getting to was just about screening the general population to catch fibrolamellars before they develop symptoms. And I think that's just really hard to put into practice because it is so rare and the kids are so healthy that, I always feel for the general pediatricians who are doing child checks once a year. There's so many things you have to try to catch that I think something like fibrolamellar probably is not going to come onto the radar until somebody actually sees a liver tumor on an ultrasound or some other imaging test.
Great. We're getting close to, I think, where we really have to call time. I wanted to share two things. We've had a number of questions from family members or patients about their specific cases, and it really isn't possible for us to answer those questions in this kind of forum. The foundation is not supposed to give medical advice, and none of you on the panel will have enough information about the individual patients. But I'd like to encourage folks that you can reach out either to some of the folks you've seen on the panel today or directly to the Fibrolamellar Cancer Foundation, and we'll be happy to connect you. You can come to our website, and we'll be very happy to refer you to specific physicians either at University of Washington or closer to where you live, who we know are quite expert in the disease, to talk about some of the upcoming treatment options and at least help you walk through the journey. And I also want to relay our thanks and the thanks of many of the panelists to all of you who've been on the panel. This has been just very, I think, thought-provoking, exciting, and despite all of the difficulties, I find it a source of optimism that there's tremendous progress being made both clinically and in the lab, tremendous dedication, and things are certainly moving faster towards therapies that are going to be even more effective than some that you heard about this morning and early afternoon.
And I'm going to add to Mark's also as a signal here to end, which is, again, thank you to everybody from the University of Washington. This was excellent. For everybody who's still on, we plan to do this with other institutions along the way, as I mentioned before, so you get a sense of everybody who's working on this. I also want to thank some of the oncologists who came on, Dr. Kent and Dr. Lee. We appreciate you being on this and everything you're doing for the community. And again, to Mark's point, info@fibrofoundation.org for the questions we did not answer. That will funnel to Lynn O'Malley, who then can help to disseminate to the right people at University of Washington or to Dr. Firth or others who may be able to direct you to better answer your questions. So we're not leaving you dry. We're not being able to answer all the questions right now. Please continue to communicate with us as much as you can. Appreciate it. Thank you, everybody.
And just to all the fibro fighters out there, keep on fighting.
That's right. Keep on tucking, as we say.
Keep on tucking.
Thanks, everybody.
Thanks all. Be well.
Thank you.